We get experimental science. We're curious, non-judgmental.
The new species of the week. I'm excited to tell you all about this new species. It is a creature. We actually have not looked at any kind of creature like this in the past. I'm very excited to highlight it. So here is the image. So I realize this is always a sneaky image of us using it, zooming in, but let's talk a little bit about this. So this is a brand new species found in mud. It's considered a new species from the South China Sea. Is the clam. It's a very thin clam, and it is related to this razor clam, it's called. So this is a razor clam, and actually what's I think really neat is Corn Relish pointed out that these things are supposedly very, very tasty. I don't actually know, Corn Relish, if I have eaten any of these. I just never knew the species. The outside is a very razor thin creature. They're called jackknife clams because of their shapes. And they're, again, considered highly delectable and very delicious. Just in general, I don't know about this particular one, but that was one component. So how did they identify this new species? It's actually a weird story, y'all. They found this shell in the South China Sea, and they're like, this looks really weird. And it was the actual researchers this time. It wasn't like the fishermen like we've talked about before. It was researchers found this shell, and all of a sudden go, wow, this is a weird looking thing. And they went and started comparing this shell to other shells that they had. Not really in a good database, but rather samples at a museum. Apparently in the museum that they were comparing against, there's a bunch of drawers with other shells, and they are just like, they're looking at their shell and they're comparing it to these existing shells. And they're just going back and forth trying to identify like, oh, what's the best match? What was really interesting about these creatures is that they looked very unique. And they thought, they were like, oh, we're going to take a look at it and try to identify if anything else looks similar. And they did find a similar shell. And here's the really cool part. The shell that they found that was identical had no match. They had no identification of it before. They had no idea what this creature was. It was just in a box. There were other samples, and no one had identified it before. It was just marked as unknown. How many shells did they find? You mean of like the original sample? So the ocean one, they found many samples. They did sequencing on 10 different shells, and their identification was actually really interesting. The part that I had zoomed in for y'all here was actually the method that they identified it. So here are a bunch of the identification bars. So there's this one here. This part, y'all, is kind of where I honed in. I was like, huh, that looks really weird. And that's in fact one of the parts. So this is like the connectical one. Yeah, the hinge part with like the different color is unique to this particular creature. Like apparently the coloration and like the difference in the two types of color is unique. I didn't realize it was that of a unique component, but surely enough, it is. The other thing, I don't know if you can kind of see it here, but here, these little bubble shapes, those little bubble shapes here are actually apparently very unique. So Dacum on here, like this isn't apparently just a fluke of a one-off of this shell. These inlet bubbles, we have no idea what they're actually for, what they're a result of, presumably something in development, but they are there and they are repeatable as well. Like you constantly see what these regularly occurring patterns underneath the microscope as you're analyzing the specimen. And so I was like, huh, that's really interesting. It's like a very easily visible methodology you're looking at. Well, so Sam, you're not wrong though, right? Like it could very easily have been a yeast because of yeast bubbling and the CO2 that came from there. So this is a cross section here. So here are the two sides of it. So switch, popped open the clam, and you're seeing the two inside components. So the outside of the shell is here. Here is actually like with the full-on look to it. And we are going to talk a little bit about clam biology as well. Unfortunately, in this instance, this isn't one of those studies where they looked very closely to figure out what is the life cycle of this creature. It's very much a descriptive paper in terms of the new features that are identified. It is skinny. It's called that razor shell for the reason of that very strong skinniness. So it's like even has like the visual element of why it's called its particular name, which I think is really neat to be able to see. So that's a unique component as well. Now, what all did they do in the study? So like I said, they kind of just found it on accident, and we'll take a look and see where they found it, in the South China Sea. So the red dot here on the map is where they identify where they did most of the sequencing from, and like that's the one like big preserved specimen that they are considering as like the average of what these animals look like. And then the blue dots are the other ranges where they identified this shell. So these are the other locations where they found this clam elsewhere in the South China Sea. How does friend mate with other friend? That's a great question, Mike. I don't actually know.
Do they have any of the internal specimen for this? Like the shells that they're... Oh, they do. Okay.
Yep.
All right.
So this image just has, they split it open, imaged both sides, broke it as well, then like imaged again both sides, just like getting the appropriate measurements on it.
Yeah.
The big thing that they're showing here that's the identifying feature are these little bubbles. Bubble-looking like structures are the thing that they were like, this is very unique to this.
So that's significant. That's not just
No.
random.
I thought it was just like whatever, like.
Okay.
But no, that is apparently significant. Like that is a repeating feature of these shells.
Okay.
They're also elongated and slender is another one of those features. So these switch are not bubbles. It's apparently a repetitive structure that they identify. So it's not just like these random set of bubbles that are appearing, these little white dots, but they are in fact a repeating feature of these shells.
Okay, one more question. When you say repeating feature, is it like they're in a certain location or just the fact that they have these weird spots?
They seem to be in a certain location. There is some movement within there, but it seemed to be like there's this straight pattern of like, there's this large bubble in this like quadrant or whatever, and then there's like next one and there's next one, like where it's like here, like it's in this zone where you'll have a big dot, and in this area you have a different kind of dot, and it just like keeps going along that front. So like it's pretty repetitive. The outside of the shell is here. This is the top side of it, and you see like the dot here is also on this side. So if it was just, again, they make the argument in the paper that if it was just a bubble, it would only be visible from one side if it was like some kind of like weird fluke, but it's visible from both sides, and it's not a function of it being super thin.
Yeah, it is so similar to the inside. That's what I was saying. I have like no perspective if it's convex or concave.
It is apparently not translucent, sloppy. I mean, at least based on their description, they do not say it as a translucent material. They're just...
Well, it has to be partially translucent because I can see color on the... All right, I'm not going to...
At least they don't say it. At least they don't say
That's
that it
fine.
is.
I mean, I imagine by the time the animal... Like, it's opaque adjacent. I'll give them that.
Yeah.
It's opaque adjacent.
See, they also... They do some genetics on this, but Lita, guess what kind of genetics they do?
Don't tell me mitochondrial. Get out of here. Really? Mitochondrial? Come on.
They do they do some nuclear sequencing too, but it's just like certain regions. So it's those single nucleotide polymorphisms. So it's just that region. And Smikes, they didn't amplify the entire mitochondrial genome. They just amplified a gene, the CO1 region.
Yeah, that one single mitochondrial gene.
That is like, I don't know why, but it's like the gold alleged standard.
It's not the gold standard, but it's what everybody does. The gold standard, meaning, the thing we should all aspire to, whole genome sequencing.
Okay, that's fair.
Period. Yeah. End of discussion.
Yeah, that's, okay, you're right. Sorry, gold standard in the sense that it seems to be what everyone does, and it seems to be the minimum threshold that everyone does if they even have any molecular stuff in there. So they did some sequencing, barely, right, for the study.
So to reiterate, they did that one gene in the mitochondria and that seems like everybody does.
Yeah, and then they did a few in the nuclear that have just been as metrics to look at for different clams.
Okay, well, that's interesting. So, I appreciate the effort.
Yeah.
That's a little bit of something.
So it's a little more than just that. See, here's more of the bubbles in different specimens, and there's different preservation types that change the outside.
Oh wait, okay, is that why it looks is E?
What it naturally is out of the water versus like when it's been.
Okay, that makes more sense.
Yeah.
Like depigmented or whatever.
Yeah, exactly.
Okay.
And then, they did this weird analysis where they did all these measurements and they put it into a principal component analysis to compare with neighboring genera that have been identified, and they showed like they were clustering differently. I don't know if that's an appropriate use of a PCA, but they did it and they showed that they were different. That's not it, but it's just a different...
What did they use for the PCA?
Like morphology measurements.
You can use anything for a PCA if If it's meaningful is a whole different question.
So that's news story number 1. It's a clam. Why did I think this was cool? We haven't ever had one.
We haven't had a clam, yeah.
I thought the story was neat about how they went back to the museum and found the same shells. They just hadn't been identified before. So it kind of was, but they had like the living samples so they can really truly get the identification. I don't know, I thought it was kind of cool.
The measurements that they did, how different are they? Because you said there's those little dots. The little dots, is that the main distinguishing feature, or like what were the cool that really set it apart? Yeah.
Yeah, so the dots were an important component of it. They had shell measurements as well, and there were differences in like the lengths and widths of the shells. Like, and the way they measured it was like the right and left valves, and like they had a bunch of different measurements on those, and those were different as well. So, and then here they mention
well, I know different, but like
the dots were the big kicker.
Okay.
The dots were the big kicker. There were worse. It seemed like they claimed statistically significant differences on the lengths.
Yeah.
When comparing them to close relatives in the area, but like to me it was those dots, because that's even in the interview they were like, the way that we identified that this was different, that we thought there was something here was the dots.
Polka dots. Yeah. Okay. All right.